Okay, hi everybody, I'm Hania Nizami. I'm going to be presenting both plasma lipoproteins 1 and plasma lipoproteins 2. And obviously the second one I think will be a little bit shorter, I'm hoping for, because it's going to be a little bit of review. Just so you guys know, the way that I design my sessions is that I try to make it as interactive as possible because I know this can be pretty long and boring. And so what I do is that I explain stuff usually using a diagram, but I might just use text sometimes or say it verbally first in a slide. And then on the next couple slides, I'm going to ask you guys questions. And so I'd really appreciate it if you guys could answer in the chat box because I have the chat box open. And so I can see if you guys are answering my questions and I can see if you guys are keeping up with me. Also, if you guys have questions just in general, if you guys want me to re-explain something or anything like that, I prefer if you do it while I'm talking or while I'm explaining. I prefer if you put the chat box then, because that way I can get the misconception just cleared right out of the way. But I'll also have some time at the end to make sure that you guys can ask questions if you need any clarification. Okay, so yeah, as you can see, I used the same PowerPoint, but I modified it and I edited it, but I didn't remove any information or anything like that, so it's gonna have everything. Okay, so now we can start. Okay, these are just the learning outcomes and I'm gonna emphasize on them like throughout the lecture. Okay, so before we start, I just want to clear some things up and I want to make sure that everybody is on the same page. So I want you guys to look at the structure of like triglyceride, and I just want you guys to be vaguely familiar with the idea that it looks like this, sort of like a letter E, because I'm going to have a mnemonic later on and it's going to use this. So as you can see, there's the glycerol over here, and it has an ester bond and it attaches to 3 fatty acids and it sort of creates this letter E formation. And then on the other hand, I have cholesterol, which has what we call like a ring formation. You don't need to draw these or you don't need to know them in detail, but I just want you guys to be familiar because I'm going to be using these drawings. Like similar to these. I also want you guys to, the reason I'm studying up with this or really emphasizing on this is that once we get into the disorders part, it's going to be so important to differentiate between triglycerides and cholesterol. Now that you guys have entered medicine, it's not enough just to know hyperlipidemia. It's not just enough to know that you have high lipids. When you guys are watching this lecture, I hope you guys are paying extra close attention to if it's the triglycerides that are elevated or if it's the cholesterol that is elevated, because these are going to look a little differently and they're caused by different things as well. So I'm going to be making sure to emphasize on that so you guys don't miss anything in the exam. Okay, so the title of this lecture is obviously lipoproteins, but I think we should go over what a lipoprotein is and how the structure is formed. So as you can see over here, I've like done my own diagram, and this outer part, so let's break it down into the outer part, which is over here, and then the inner part, which is inside the blue circle. Okay, so there's 2 parts of the lipoprotein. There's the outer part over here, and then there's the inner part. The outer part, what I want you to notice is that it has these 3 things. It has phospholipids, which I've drawn for you like this over here. It has free cholesterol. Notice I'm saying free cholesterol. This cholesterol has not been esterified. Esterified means that there's been a fatty acid attached to it. This cholesterol has no fatty acid, it's just free. So the free cholesterol is on the outside, okay? The free cholesterol is on the outside. I also have my phospholipids. And I really want you guys to notice this purple circle that I've drawn over here, because this thing is called an apolipoprotein. It's essentially a protein that's stuck on the outside of my lipoprotein. Okay, so my apolipoprotein is a protein and it's stuck on the outside of this entire structure, which is called a lipoprotein. And it's really important because later on in the lecture, I'm going to be differentiating between the different types of apolipoproteins. There's like multiple forms of them. And I don't want you guys to get confused between an apolipoprotein and a lipoprotein. So an apolipoprotein is just the protein, and the lipoprotein is this entire structure over here, okay? So I told you what was on the outside of the lipoprotein, right? I told you there's a phospholipid, just monolayer, just one layer. I told you that there's this free cholesterol. It does not have a fatty acid attached to it. It's a free cholesterol. And I told you that there's an apolipoprotein, okay? The function of my lipoprotein is to transport lipids, okay? So on the inside of my drawing, I've drawn two main structures. So the two main types of fats that we're going to try to transport in lipoproteins, they're either going to be triglycerides, which I've drawn for you over here, which I told you looks kind of like an E. This is my glycerol, and it's attached to 3 fatty acids. So my lipoprotein is either going to carry triglycerides, which are on the inside, or it's going to carry esterified cholesterol, right? Cholesterol esters. So I drew this little fatty acid attached to my cholesterol to make you guys remember that the free cholesterol is in my membrane, but the esterified cholesterol is on the inside. And we call this inside part the lipid core. And the reason it's called lipid core, obviously, is because it has 2 types of lipids. It has my triglycerides, and it also has my cholesterol esters. So we have 3 things that make up the outside, and we have 2 things that make up the inside of my lipoprotein. I think this should go without saying, but obviously the outside part is going to be more polar, because when I think about my lipoproteins, I told you that their function is to transport lipids. So in order to transport lipids, the outside part has to be polar so that it can dissolve in water, because otherwise if it's non-polar, it's just going to it's not going to dissolve in the water, and therefore it can't transport the lipids that are on the inside. Okay, there's one more thing I want to emphasize on, which is that the bonds within a molecule, so for example, my fatty acid to my glycerol, my fatty acid to my cholesterol, even my fatty acid to my phosphate, all of those, we call those covalent bonds. Those are very, very strong. They share electrons, covalent bonds. However, the bonds between my lipids and, for example, my proteins, right? Like the bonds that are keeping this lipids on the inside are non-covalent bonds. They're not sharing electrons. What keeps these lipids on the inside is non-covalent bonds, okay? But within a molecule, I'm going to have covalent bonds. I'm going to repeat this again throughout the lecture, so if it doesn't make sense now, that's okay. Okay, so let's just go over briefly. I kind of already talked about this, but the function of the lipoprotein, like I said, is that it's going to deliver the lipids and maybe deliver them okay, so somebody that said they didn't get the covalent bond. So a covalent bond is when I share, but between 2 molecules when they share an electron. It's just what I want you to understand is that there are 2 types of bonds. There are covalent bonds and there are non-covalent bonds. Covalent bonds are very, very strong, and they're literally what changes a molecule structure. So if I add a fatty acid to a cholesterol, I'm adding a covalent bond, and it literally changes the chemical structure of my cholesterol. I no longer call this cholesterol. I call this cholesterol ester. However, for example, between my cholesterol ester and my triglyceride, between these 2 things, notice I didn't draw anything connecting them, but there has to be something weak. There has to be something that's at least keeping them together inside this lipid core. And those are non-covalent bonds. Those are weaker, and they're called non-covalent bonds. So between the different molecules, like here, even proteins, mostly actually it's the proteins and the lipids, this is non-covalent bonds, and they're just keeping the lipids on the inside. But within a molecule, I have covalent bonds. Does that make sense now? Yeah, okay, awesome. Okay, yeah, so this slide, okay, yeah, so we're going to be going over this in more detail later on, but I think what they just want you to know is that the general function of a lipoprotein is that they are required for giving, so for pretty much when I'm exercising, when And when I'm doing anything, honestly, there's going to be parts of my body, certain cells in my body that are going to need lipids. They're going to need lipids for multiple reasons, maybe for energy, maybe as a structural part. I'll go into their specific functions later, but we just need lipids in different parts of our body. So the point of a lipoprotein is that it packages the lipids on the inside and it delivers it to all these parts in our body. I also want you guys to know that lipoproteins also can go back to the liver. And again, I'll be emphasizing which lipoproteins, for example, go back to the liver and which don't. But yeah, you just have to understand that like once my lipoproteins go to my cell, they're going to go through this process of endocytosis. Endocytosis means my cell is going to take in like the lipoprotein, it's going to start to degrade it, and it's going to start to use the lipids for its own benefit. Like I said, it could be storage, not storage, it could be for energy, it could be for structural units. So I think that's the main thing they want to go over. This top point I'll go over later on because it's more relevant then. Okay, so here's my first example of me quizzing you. So I told you the structure of a lipoprotein, and I told you that there's on the inside, right, there's a something core. What is that something? What do I call the inside of my lipoproteins? Yes, exactly. Thank you. It's called a lipid core. Yeah, and it's obviously the non-polar part, and I have to keep the outside of it polar in order for it to dissolve. Okay, and I talked about how there were specifically two types of lipids on the inside of the lipid core. We had my triglycerides and we also had my cholesterol esters. I told you that the polar portions were on the surface in order to make them soluble. Here's the last point that I'm going to explain, and I'm going to test you on this later on, so be sure to be listening. Okay, so in order to get lipids, in order to get these triglycerides and my cholesterol, I can either get them from my outside of my body or the inside of my body. When I get them from the outside of my body, I call these dietary lipids. Okay, so I just told you that I can either get lipids from the outside, which is exogenous, or on the inside, which is endogenous. If it's from the outside, I'm going to call them dietary lipids. From the inside of my body, they can either be from adipose tissue or they can be from my liver. Okay, so if they're coming from the inside of my body, they can either be from my fat cells or they can be from my liver. Now, the liver is really cool, and this is something I want you guys to remember. My liver does not only store lipids, it can also make lipids, literally from scratch. Your liver can make lipids. So just again, I have sources of my lipids. I can either get them from the outside. If it's from the outside, I call them dietary lipids. If I want to get from the inside of my body, I can either do it from adipose tissue or I can do it from my liver. My liver, you have to remember, there's two ways. It's either going to be the storage form of the lipids or my liver can just make the lipids from scratch. Okay, and I'll repeat this again later on. Okay, so this is another thing that I want you guys to understand, which is that when I have my free fatty acid just in this form, it's going to be able to bind to albumin and just be transported like that. However, if I form an ester bond, okay, with my fatty acid, so for example, over here, look, I have my fatty acid and it formed an ester bond with my glycerol, okay? Once this happens and I form a triglyceride, then I need to package this inside of my lipoprotein. Same thing happens when I add an ester bond. Ester bond's like somewhere here on my cholesterol and making a cholesterol ester. I need to put my triglycerides and my cholesterol esters into lipoproteins. However, my free fatty acid, before it forms the ester bonds, my free fatty acid can just bind to albumin free in the blood. Okay, so my unesterified free fatty acid can just bind to albumin in the blood. Okay, so exactly what I just said in the previous slide, I'm going to be testing you on. So is it the esterified form of my free fatty acids, or is it the unesterified form of my free fatty acids that are transported by serum albumin? Yes, non-esterified, good. Exactly, yeah. And then this is a repeat of something that I said earlier. I told you that the lipids that are in the center of my lipid core, they form a specific kind of bond with the proteins. Was it covalent or was it non-covalent? Yeah. Okay, I see one answer. Do more people want to answer? Because I like when more people answer. But that's right, it's a non-covalent bond. Okay, and this is a repeat, and I think my thing got messed up. But I told you that when you think of lipid sources, I need you to think of dietary lipids as the outside source, and then the inside source, I have adipose tissue, or I have liver, like my liver also stores some lipids. Okay, this is a really important concept, and I kind of touched on it before, but I really want you guys to understand this. When I say reverse transport of cholesterol, when I say that the cholesterol is going from certain peripheral tissues and they're going back to the organ, which organ am I talking about? Yes, the liver. When I say reverse transport of cholesterol, I'm saying that my cholesterol is going from my peripheral tissues and it's coming back to the liver. Okay, so I would say all of that was pretty easy, pretty straightforward. You've been keeping up, that's pretty good. We're going to go into more of the specifics now, and I'm going to be classifying the lipoproteins specifically, and I'm going to tell you how these things are different, and especially I want you guys to know how to recognize them, for example, in disorders, because I think they really like to test on that in exams. And just making sure that you guys don't miss anything. So if you look over here, I've inputted a diagram that shows you the different sizes, or rather the different densities of each type of lipoprotein. So what I want you to notice is that as the lipoprotein gets smaller, the density gets higher, okay? So I'm gonna say that again. As the lipoprotein gets smaller, the density gets higher, right? Like I'm making this more compact. And what I want you to understand is that if I have a very small lipoprotein, it means that it doesn't have a lot of lipids on the inside. There's no way there's going to be a lot of lipids on the inside. I don't know if it gets my mouth, but I don't know if there's not going to be a lot of lipids on the inside of this very small molecule. And that's why the smaller sort of molecule, or rather the smallest lipid protein, is what we reference as the healthy one, because it's so small, it doesn't really have a lot of lipids in it. Rather, it has a lot of proteins. And if we compare lipids to proteins, we would prefer to have a lipoprotein in our body that's high in protein rather one that's high in lipids. So again, because this is very small, HDL is very small, it cannot carry a lot of lipids. And because it's not carrying a lot of lipids, it's considered the healthy one, because lipids are bad for us, right? Okay, so I want you guys to look at the order of this again. I'm going to make you memorize it, but this really giant, huge one over here is called my chylomicron, and I'm going to be going over how the chylomicron is metabolized. But something I want you to know about a chylomicron, I'm going to put the laser pointer on 1 second. Yeah, my chylomicron is that majority of my chylomicron carries triglycerides. So remember how earlier on in the beginning, I told you you have to know, you have to know the difference between triglyceride and cholesterol. You have to know that these are two different types of lipids, okay? So my chylomicron mostly has, like literally 90% has triglycerides, okay? And then my VLDL also has quite a bit of triglycerides. There is triglycerides in these ones, but mostly these ones are made of cholesterol. So just again, these two really giant ones are mostly triglycerides, chylomicron and VLDL, versus the smaller ones are mostly cholesterol. Keep in mind that now that you're in medicine, things work in shades of gray. There is going to be some cholesterol in chylomicron. Yes. There is going to be some triglycerides in HDL. Yes. But mostly, percentage-wise, the two very giant ones are triglycerides, and the smaller ones are cholesterol. And I'm going to have a mnemonic at the end for you guys to remember that. Okay, so since I spent so long on that, this is the order of the size. Okay, so you guys tell me, what is the largest lipoprotein and what is the smallest lipoprotein? Or first, tell me what the largest one is. Largest, yeah, is chylomicron. Good, yes. And the smallest one is, yes. So I think this is my fault because I asked both, so I'm seeing some mixed ones. But largest is chylomicron, largest. I'm not talking about density, I'm saying literal size. And the smallest is HDLs. This works the opposite when I talk about density, right? I told you that they're inverse. Chylomicrons are the least dense versus high density, literally from their name, are the most dense. Okay. And those are really easy so far, guys, but I just want to make sure that your base is strong. Okay, so this is the mnemonic that I talked about. For now, ignore the cholesterol one. I'm going to be going over that in my second presentation, but let's look at the triglycerides. I told you that triglycerides sort of look like an E to me. Hopefully everyone can see that, right? Like the glycerol and then the fatty acids, it sort of looks like an E. So let's say that the mnemonic for triglycerides is that you want to remember the structure of it and that it looks like the letter E. When you hear E, I want you to think of eat, okay? Triglycerides, like the way that you eat food. And the reason I want you to remember that is that the majority of our triglycerides actually are obtained from our diet, specifically from chylomicrons. I know you guys have done the GI block, so I know that you guys actually learned that. And the way that we absorb triglycerides is from the GI tract, specifically from the small intestine, right? So it looks like the letter E. When you hear E, I want you to think eat, because triglycerides are mostly obtained from our diet. Specifically, we absorb them from the small intestine to the liver using chylomicrons, okay? The other thing that I want you to remember with E is that when you eat, you get bigger. And that's why the bigger lipoproteins, specifically chylomicrons and VLDL, okay? Specifically chylomicrons and VLDL, these ones are big because when you eat, you get bigger. And so that's why these two carry triglycerides, okay? Mostly carry triglycerides. And then the last thing, remember how I told you in the beginning that on my lipoprotein, I have these small proteins on the outside called apolipoproteins. And I told you that there are different types of apolipoproteins and that it's really important to know which ones are located on which lipoprotein. So what I want you to remember is that when you see E, I want you to think eat, and I want you to think of food. And I also want you to think of ate, because when you hear ate, you know, the past tense of eating ate, you're going to think ApoB48. ApoB48, because ApoB48 is only located on chylomicrons. ApoB48 is only located on chylomicrons, and the way that you're going to remember that is that I just told you that chylomicrons are responsible. They're part of our diet, right? They're responsible of transporting the dietary triglycerides. So if you remember ApoB48, it should make you think of chylomicrons, ate, like I ate food, so chylomicrons, transporting the lipids from our small intestine to our liver. And that should help you remember that ApoB48 only on chylomicrons, okay? I'm gonna test you on that later on. I'm really gonna nail that idea into you guys. So hopefully you guys are paying attention. And again, don't worry about this one right now. I'll explain it in the second presentation. Okay, so chylomicrons. I emphasized on this already 100 times. Chylomicrons, which type of lipid is inside of a chylomicron? Is it triglyceride or is it cholesterol? Yes, triglycerides. Okay, thank you guys. And it's the highest percentage of triglycerides. If you look at the diagram, like it's so huge. Look at how much this soft yellow color, that's triglycerides. And the chylomicron, like it just has so much of that. Okay, and then I told you that it gets successfully denser as we move down over here. HL smallest and the densest. So I don't know if this is necessary, but because you guys have already answered this, but could you guys just put in the chat the order? What is the lowest density? I'm not saying size, I'm saying lowest density, and then to highest density. Yeah, so the first one is right. It goes chylomicrons, VLDL, LDL, HDL. Yeah, okay, yeah, you did the greater than sign. Okay, that makes sense. So yeah, CVLH, yeah, that works. Exactly, so lowest density chylomicrons, highest density HDL. Okay, good, you guys are keeping up. Okay, so this is okay. So when we talk about, actually, let me go back over here. Okay, so this is where things get a little bit confusing, but you guys have been doing really well so far, so I think you'll be able to understand. If I show you this diagram, let me just move my chat box 1 second. If I show you this diagram, I know I told you VLDL is responsible for transporting triglycerides. This is true. VLDL is responsible for transporting triglycerides, but you guys have to think in shades of gray. You guys have to think in terms of medicine. It still has cholesterol, right? Just because I'm telling you that the function of VLDL is to transport triglycerides, don't think that it's purely made out of triglycerides. It still has some cholesterol. It's just that VLDL has more triglycerides than it does cholesterol. I'm going to contrast this to something like chylomicrons. Chylomicrons, again, they do have a little bit of cholesterol, that's true, but the chylomicrons are literally 90% triglycerides, that when we calculate the cholesterol in our body, we ignore the cholesterol that's inside of chylomicrons because it's so small inside of chylomicrons. Chylomicrons are almost exclusively triglycerides. However, on the other three, all of them carry enough cholesterol, especially HDL and LDL, actually, especially LDL. They carry enough cholesterol that we want to include them, including VLDL. Yes, VLDL has more triglycerides, but it has enough cholesterol that we still want to include it in our calculation of cholesterol. So what I'm trying to say is that when we calculate cholesterol, we include all lipoproteins in our body except for chylomicrons, because chylomicrons are 90% triglycerides. We don't really care about the cholesterol that's inside of them. It's so small, we don't care. Okay, so which 3 lipoproteins, you can say 3, you can even say 4, lipoproteins make up the cholesterol in our body that we actually calculate? Which are the 3 that I told you? Yeah, VLDL, LDL, VLDL, LDL, HDL. I think somebody mentioned IDL as well. And I think that should be valid as well, in my opinion. It's just that I don't know why, but nobody really cares about IDL. I barely see it referenced in textbooks or whatever. But for sure, you should remember that VLDL, HDL, and LDL, IDL should be considered valid as well. And all they're trying to tell you with this, I don't think you guys have to memorize the ranges in terms of first year, but they're just trying to convey the concept to you guys that we have a maximum limit that we want our cholesterol to reach. We don't want the cholesterol to raise above a certain level. And I'm going to mention why later on. There's a very specific side effect that we're very scared of regarding cholesterol specifically. Okay, and then let's go on to the other parts. So this bullet point over here is very important for when we get to the disorders, but elevations of plasma triglycerides are usually caused by increased VLDL. I told you about this, right? I told you VLDL, think triglycerides. It carries mostly triglycerides. What else could you think of? What other lipoprotein could you think of that if I had too much of this lipoprotein, it would be too much triglycerides? Yes, chylomicrons. Okay. Good job. I underlined usually over here because too many chylomicrons is very rare. So usually the cause of too many triglycerides is going to be VLDL. Okay, that's why. But if you had too many chylomicrons, it would still lead to it would lead to elevated triglyceride levels. Okay. And then I also said over here that shouldn't it be primarily chylomicrons? Okay, I totally get what you're saying in terms of the percentage of triglycerides. What I said that usually, I'm not talking about the molecule, I'm talking about the percentage of the population. A deficiency in chylomicron breakdown is so rare compared to a deficiency in VLDL breakdown or something that causes too much VLDL. Like too many chylomicrons just in the population is just such a rare disease that's why she said that usually if you see a patient and they have too many triglycerides, you're going to assume it's because of VLDL, just because too many chylomicrons is very rare. Like that disorder is very rare. Does that make sense? Okay, yeah. And then I told you that like when we talk about too high of cholesterol, the main thing that we're worried about is too high of LDL, because I told you that LDL is the one that carries the cholesterol. At the bottom over here, I said, I'm going to explain to you why too high levels of HDL would not lead to elevated levels of cholesterol. Okay, somebody said not enough cholesterol in HDL, it's mostly protein. So this is good thinking, but this isn't what I was trying to get at. It's because HDL is actually good for us and it transports the cholesterol from the peripheral tissue back into our liver. In other words, or actually even the cholesterol that's in our blood, it transports that back to our liver. So it actually lowers cholesterol. It does not raise cholesterol levels. It actually lowers cholesterol levels. I'm going to explain that again later on in way more detail, so it doesn't make sense now. That's okay. Okay, so over here, what they're trying to tell you, and again, you guys have learned this in GI, and I've drawn this in another diagram, is that in my liver, I have cholesterol that's being stored, right? However, when I eat some like really fatty food, I eat from McDonald's, what's going to happen is that as soon as that fatty food enters my GI tract, my liver is going to use the cholesterol and add it into the bile. And so the cholesterol that's in my liver is going to be sort of removed from my liver and being added to my GI tract. And the main idea that they're trying to tell you over here is this idea of sort of like recycling. So the cholesterol is being added to the bile acids or the bile in my liver. The bile is entering into my GI tract. And then the cholesterol that I just used to make bile is actually going to be reabsorbed again. Okay, so the cholesterol that just entered my bile and entered my GI tract is going to be reabsorbed again through a very specific lipoprotein. Do you guys remember what lipoprotein is responsible for reabsorption from the, yes, it's from the chylomicron. This apolipoprotein B48 should have given it away because there's only one lipoprotein that has ApoB48. Eight like you ate food, think chylomicrons. So this cholesterol is sort of the one, I like to think of it as it's the one that was in the liver. So it was a cholesterol that was in the liver, it went into my bile, it went into the GI tract, and now it's being reabsorbed from my chylomicron. The reason I'm trying to differentiate between that is because most of our cholesterol is from the inside of our body versus most of our triglycerides is from the outside of our body. Does that make sense? So the cholesterol technically started from the liver, not from our food. Does that make sense? What about the cholesterol that came from the food? That's also going to be absorbed. But if you compare it percentage-wise, most of our cholesterol still comes from the inside of our body. That's okay? That makes sense? Okay. Okay, so over here, I drew a little diagram for you guys that it's a little bit messy, but I just want to explain the general idea over here. So again, I have my cholesterol that was in my liver. It's entering into my intestine as bile. And then the cholesterol that was in my bile is going to be reabsorbed from my chylomicron, which mostly carries triglyceride, but it will carry a little bit of cholesterol as well. And then it's going to go back to my liver. I told you ApoB48 is specific to my chylomicron. Another concept that I want to introduce to you is that oftentimes when I try to find diagrams on this, it looks really confusing because the concept of recycling, and I don't know where to start. So I'm going to tell you guys where to start. So when I talk about VLDL, what I want you to know is that it's made by the liver and its role is to sort of distribute the fats, mostly triglycerides, although it will distribute cholesterol as well. And as my VLDL is going around in my bloodstream, it's giving the triglycerides to the peripheral tissue, it's going to be broken down smaller and smaller until it forms IDL, and then it's going to form LDL. So the main concept I want you guys to understand over here is that VLDL, IDL, LDL especially, its role is to deliver fats to the rest of my body, to extrahepatic tissue, parts of my body that are not the liver, okay? And the reason I really want you to understand that is that this is the opposite function of HDL. I told you HDL does the opposite. Its role is to get the fat that's in my peripheral tissue and it's going to bring it back to the liver. And that's why I drew these angel wings over here. Because if you think about it logically, our liver is awesome. Like it knows how to handle so much. It knows how to store so much. It can regenerate. So our liver knows how to store fats really, really well. Compared to the rest of my body, it doesn't really have good storage mechanisms. So if I compare LDL to HDL, HDL is bringing the fats back into my liver, back to this liver that knows how to store it, versus LDL is bad and it's putting the fats into places that don't really belong. Although obviously you do want a little bit of LDL, but the problem is that when it gets too much, the LDL is going to put all this cholesterol into places that don't really belong, like the peripheral tissue. Okay, I did draw these dotted lines because I want you to understand that these lipoproteins can obviously go back to the liver. It's not just this like one-way pathway, it's a cycle. But I just wanted you to understand the main function that HDL goes from peripheral tissue back to the liver versus LDL is delivering the cholesterol to the peripheral tissue. So hopefully that distinction makes sense. I will test it again later on to make sure that everyone's caught up. Okay, and I drew the apolipoproteins, but I'm going to explain them later on. Okay, so I've already nailed this into you guys that I've told you which ones are the biggest, like the order of increasing size, and I've emphasized that this is the opposite. So make sure to read your exam question carefully. This is the opposite order of increasing in density. However, there is a little bit of a caveat to this. There is a little bit of a confusing thing. If they ask you specifically for electrophoresis, this gets a little bit confusing because if you understand electrophoresis, it's actually measuring two things at once. The first thing it's measuring is size, and the second thing it's measuring is polarity. So if you look at size, it's just a simple concept. The really, really big ones, for example, my Kylomicron is so huge, it can't even move. You know what I mean? Like it's just so big that it's like immobile. So that's why it stays right at the origin. Similarly, my HDL is so small, it's so easy for it to move. So it's going to go from the origin over here and move all the way across. Like it's going to be the farthest one. However, did you guys notice that LDL and VLDL are switched when we talk about size? If I ordered this in terms of size, I would have said Kylomicron, VLDL, LDL, and then I would have put HDL. And the reason for this is that I can't use, for an electrophoresis specifically, I can't use size alone. Exactly. It's because of the polarity. VLDL, if you look at my previous diagram, right, VLDL has like way more, or at least it has more apolipoproteins. It has more proteins versus LDL only has one. LDL only has one apolipoprotein. I'm going to really nail this into you guys, don't worry, but it's ApoB100. And so it doesn't have as much polarity, right? Because it's proteins that cause polarity versus my VLDL is more polar. And so because of that, it's more polar, it's going to be more attracted to the anode. So you can imagine it's a little bit more negative, for example. So it's going to be attracted to the anode. Versus my LDL is not as polar, so it's not as attracted. And like their sizes are sort of similar, and so that's why this difference comes about. So this is very specific to electrophoresis specifically. I'm not telling you the VLDL is smaller. That's not what I'm saying. I'm saying it's more polar. So hopefully that makes sense. Okay, so now we are going to talk about the apolipoproteins. Oh, what's up? Okay, somebody said to repeat, please. Okay, let me just go back. Okay, so doesn't polarity increase as we go down in size? So you're saying like the more dense that it is, the more polar it is. I can understand like your logic with that, but I think what's more significant, or the way that this was explained to me at least, is that the main factor, more protein in terms of like ratio. That is true. There is a higher protein to lipid ratio, but I'm talking about like literally like with VLDL, I have literally more protein, not in terms of ratio, but I literally have extra proteins on my VLDL compared to my LDL, which just has one protein. So even though you're right about the ratio, I think the ratio is not that significant compared to VLDL to LDL. What's more significant is that I have extra proteins, and more proteins mean more polarity. Like amount, you mean? Yeah, literally like VLDL has 3 proteins, for example. LDL only has 1 protein. Like literally the number of proteins is different, not the ratio. Well, the ratio is also different. But does that make sense? Like what I care about with polarity is that I care about the number of proteins, not the ratio. Even though your thinking is very valid, but it's just that like when I do an experiment, this is what I care about. So does that make sense? Can I move on? Okay, so just be sure that when you're reading the exam question, you make sure to read, is it an electrophoresis or is it not? Because if they're asking about gel electrophoresis, just keep in mind that I have to keep in mind the polarity and specifically the number of proteins. Okay, so now that we got that out of the way, I'm going to be going into specifically the different types of apolipoproteins, and I'm going to be sure that everyone who leaves here today understands and memorizes which apolipoproteins belong to which type of lipoprotein, you know? So let's start with the first one. The first lipoprotein, apolipoprotein I want to talk about is ApoB100. Okay, so ApoB100 is this apolipoprotein that my liver, let me just remove my chat box 1 second. So I want you to think about this almost like the liver is like the mom, like it's this mom, and it has this child that is VLDL, and it tells VLDL, or it attaches ApoB100 to the VLDL in order for the VLDL to deliver its triglycerides. So pretty much the function of ApoB100 is so that the cells that are on the peripheral of my body, not the peripheral of my body, but like the cells that are not in my liver, so my fat cells, my muscle cells, whatever, they recognize ApoB100, and so that the fat that is inside my VLDL can be delivered to my peripheral tissue. So that's the function of ApoB100. It's delivery of fats, right? It delivers the fats inside VLDL to the peripheral tissues, okay? And what I want you to know is that, so VLDL was told by its mom, the liver, to carry this ApoB100, right? And even as VLDL is broken down, even as VLDL is broken down to IDL, to LDL, it never, ever, ever loses its ApoB100, okay? So the ApoB100 protein that the liver gave it, even if the VLDL is broken down, it keeps its ApoB100 protein. Okay, and the way that I want you guys to remember that is that the liver is saying B100, so ApoB100, B100 percent prepared for your delivery. For your delivery, you should remember the function of ApoB100. It's for delivering the fats. So for example, for VLDL, it's triglycerides. For LDL, it's cholesterol. And also to come back to me. So be 100% prepared for your delivery and to come back to me. The come back to me should remind you that the ApoB100 is also useful for the hepatocytes to take up the lipoproteins. The main function I would still say is for delivery, but I don't want you guys to get confused, and I do want you guys to know that ApoB100 still can attach to a hepatocyte, and it can still cause the return of the VLDL or the IDL or the LDL back to the liver. So it sort of serves two functions, but I would argue that the delivery is much, much more important. So does that make sense? Why all three, VLDL, IDL, LDL? Okay, somebody asked a question about my chat box. Oh my God, sorry, my chat box is glitching. When it's delivered, where does the APO go? So I guess when you think about this, what's happening is that the lipoprotein is going to a cell, right? And it's like entering the cell, and then it's like being broken down, or it's being like endocytosed, and then the lysosomes are breaking it down inside of the cell. But then the ApoB1, like when the lipoprotein reaches the outside, when it goes back outside of the cell, the ApoB100 will still be attached. Like it will be recycled, essentially. Like what I'm trying to say is that like the VLDL, it's only going to deliver the lipids inside of it. It's not going to give up its ApoB100. It's just going to, the ApoB100 never detaches from the VLDL. It just stays attached to the lipoprotein, even after it enters the cell and that kind of stuff. Does that make sense? I have a diagram later on, maybe that one will make more sense. But just think that the apolipoprotein just stays attached to the lipoprotein. It's only the lipids inside that is being delivered. Can I move on? Does that make sense? Yeah? Okay. If it doesn't, I'll go over it again. Okay. Like later on, I have a different diagram. Okay, so moving on with the apolipoproteins, I have some more mnemonics, and I'll test you guys on this later on as well. So for ApoC2, what I want you guys to know is that the C is for cleaves, and the 2 is that it cleaves triglycerides into 2 parts. So ApoC2 cleaves or cuts triglycerides into 2 parts, okay? I also said over here that it activates a specific enzyme called lipoprotein lipase. So lipoprotein lipase needs ApoC2 because ApoC2 cleaves triglycerides, not cholesterol, triglycerides into 2 parts. And that should make sense because if you think about lipoprotein lipase, it's literally lipoprotein lipase. It's responsible for breaking down the lipids that are inside of a lipoprotein, specifically the triglycerides. I'm not talking about the cholesterol. Okay, this is really important because I'm going to emphasize this. Okay. Okay, yeah. And then I told you that there are only 2 lipoproteins that I said that carry triglycerides. Yes, it's in small amounts of other lipoproteins, but there are 2 lipoproteins that I wanted you to remember that have high amount of triglycerides. What were they? Yes, okay, good. Kylomicrons and VLDL, those are the 2 that I want you to remember that mostly contain triglycerides. So now, doesn't it make sense to you that the 2 lipoproteins that will have ApoC2 are going to be chylomicrons and VLDL because the other ones aren't as involved. They don't have as much triglycerides, so they don't need ApoC2 because ApoC2 specifically breaks down triglycerides into 2 parts. I put another mnemonic over here, which is that ApoC3. You don't need to know where ApoC3 is found. You just need to know that ApoC3 leaves the triglycerides to be. So what I'm trying to say is that it leaves the triglycerides alone because it's actually going to inhibit LPL. So it's going to prevent the breakdown of triglycerides by inhibiting LPL. So ApoC3 leaves the triglycerides to be. It prevents the breakdown of triglycerides. Because it inhibits lipoprotein lipase, is the opposite of ApoC2. C2, C for cleaves, 2 for 2 parts, specifically triglycerides. Hopefully I really nailed that into you. Again, I will test you on it again. I already told you guys about ApoB48. The 8 should remind you that you ate food. If you ate food, then you should think of chylomicrons because chylomicrons are related to dietary triglycerides or dietary lipids. The last one that I want you guys to remember is ApoE, which essentially what the function of ApoE is that if you guys remember on your liver, I have these receptors that are called LDL receptors. They're not called VLDL receptors. They're not called HDL receptors. They're called LDL receptors. And so the problem with this is that the hepatocytes recognize LDL by itself, doesn't need any further modification. But all of the other lipoproteins need ApoE because they need to trick the LDL receptor that's in the liver into thinking that like, oh, I'm LDL as well, like take me in, please endocytose me, you know? But like LDL doesn't need it because LDL by itself can already attach to the LDL receptor. But all the other type of lipoproteins, they need ApoE, otherwise they can't attach to the LDL receptor. Like all of them attach, all of the lipoproteins, all of them go to like the LDL receptor. But the difference is that the ones that aren't LDL, everything but LDL, they need the ApoE. Okay, does that make sense? So ApoE is needed for everyone else but LDL because LDL can automatically attach to the LDL receptor. Does that make sense? Can I move on? Okay. Okay, and then I believe this is my last mnemonic regarding the apolipoproteins, and then the rest of this will mostly be review. But I have that Hadil, HDL, always gets the high scores. She always gets A's. The A's should remind you that when I talk about capital A lipoprotein, when I talk about capital A apolipoproteins, so apolipoprotein A, they're only found on HDL. Any of the other lipoproteins, they do not contain apolipoprotein A. Okay. And that this is why she was the only one that passed her law college admission test, LCAT. The LCAT should remind you that the LCAT enzyme only works with HDL. So the function of the LCAT enzyme is that it converts the cholesterol that's in the peripheral tissue to cholesterol ester, and this allows for the cholesterol to move from the peripheral tissue to the liver. I told you that HDL is the only one that does this. HDL is the only lipoprotein that moves the cholesterol from my peripheral tissue back into my liver. So that's why HDL is the only one that interacts with LCAT. LCAT doesn't stand for Law College Admission Test. It stands for something else, the lecithin cholesterol isyltransferase, but it's an enzyme, and the function of the enzyme is to convert the cholesterol so that it can move from the peripheral tissue back into the liver. Okay, so there's two main things I want you to remember from HDL. HDL is the only one that has apolipoprotein A. Okay, all the other ones will have apolipoprotein B48, B100, whatever. But apolipoprotein A, especially like A1, you have to think HDL. The other thing is that the LCAT enzyme, it only interacts with HDL because its function is from peripheral to liver, and the only lipoprotein that I told you that did that was HDL. Okay, so this is the mnemonic. Hopefully it helped. Okay, yeah, so now to quiz you guys, the Kylomicrons, there was a specific apolipoprotein that I've said at least 3 times now that is only present on Kylomicrons. Which one was it? Okay, I see one answer. Does anybody else want to answer? Good. ApoB48 is located on Kylomicrons. I showed you that diagram of the VLDL that was getting broken down and was told by its mama, the liver, to go do a delivery. And there was a specific apolipoprotein that I told you is present on VLDL and ALDL. ApoB100, yes, ApoB100. The other thing that you can think to remember this is that if you think of the first 3 letters of liver, LIV, it should remind you that L for LDL, I for IDL, V for VLDL. These 3 are the 3 lipoproteins that are made by the liver. And because they're being made by the liver is telling them, be 100% prepared for your delivery. And so it's giving them ApoB100. So again, it's the first 3 letters of the word liver, LIV, LDL, IDL, VLDL. Okay, and I told you that the HDL, it has, okay, somebody already answered. Let's have other people answer. The HDL, it contains a specific kind of apolipoproteins only found in HDL. Yes, apolipoprotein A. Good job, guys. Okay, and so these are the functions of apolipoproteins. I told you that there was an apolipoprotein that regulated an enzyme in the blood. For example, specifically breaking down the lipids that are that are found in lipoproteins. Which apolipoprotein activated that enzyme? Okay, I see one answer. Maybe I should phrase this better, but like there was an enzyme that I told you guys about. Okay, I'll tell you the name. It's lipoprotein lipase. Yeah. And there was a specific apolipoprotein that I wanted you guys to know about, and you guys are saying it correctly, ApoC2. ApoC2, C for cleaves 2 into 2 parts. It cleaves triglycerides, not cholesterol, don't get it wrong in the exam, triglycerides into 2 parts by activating lipoprotein lipase. Exactly. Okay, let's jump to this third one, and then I'll go back to the second one later. I told you before that the function of a specific apolipoprotein was so that the peripheral tissue would recognize these lipoproteins so that they could take in the lipids. They could endocytose the lipids. So the function of this apolipoprotein was for delivery of lipids, right? So they could be endocytosed. Okay, some people are saying ApoE. ApoE is also correct. That is true. It is also for recognition. For recognition from the liver specifically. So that's also a good one. What about another example of an apolipoprotein that causes endocytosis, but not in the liver, in the peripheral tissue? So ApoE is for the liver. There was another one that I told you that was for delivery. Yes, ApoB100. I told you that the function of ApoB100 was so that the peripheral tissues recognize VLDL, IDL, LDL, and so that they could endocytose those lipoproteins in order to take in those lipids. Does that make sense? So ApoE, think liver, to recognize for the liver, and ApoB100 generally is for the other parts of the body. And then just the second one, I haven't talked about this yet, but the second function is to transfer the lipids, right? I'm going to go into detail about this later, but you guys should know this protein called CETP, and it gives the cholesterol that's in HDL to other lipoproteins. So it could be VLDL, it could be LDL, but CETP, it delivers the cholesterol that's inside the HDL, and it's going to give it to other lipoproteins, such as VLDL or LDL. I'm going to ask you about that again later, so if you didn't get it this time, it's okay. So this is kind of an interesting idea, but if you guys have ever wondered like where these names come from, like why is this called ApoB48 and why is this called ApoB100? It's actually, is CETP harmful? Okay, we'll go into CETP more later, but that's a very, very good idea. Is CETP harmful? And the short answer is yes. But yeah, exactly, alternative splicing. And the interesting idea is like, why 48? Like, how on earth did they come up with 48? And it's because if I look at this ApoB protein, the 48 is because this protein has like 48% of the original protein. You know what I mean? Like, imagine I have an apolipoprotein B, and if I take 48% of that protein, then it would look like ApoB48. Versus ApoB100, it's just 100%. It's the entire protein. So ApoB100 would be bigger. That's just like an interesting idea. And they just want you to understand that it's like there's this one common gene that makes ApoB, that protein, like the ApoB protein. And depending on how I do the alternative splicing, and depending on how I decide to translate this, I can either get ApoB48 or ApoB100. So I think they just want you to know it's the same protein. It's just been like spliced differently. Okay, so I already introduced the idea of lipoprotein lipase to you, and you guys already knew that it was ApoC2, right, that cleaves triglycerides into two parts because it activates lipoprotein lipase. As you can see over here, lipoprotein lipase is actually not inside of the cell. It can be, well, it depends, but sometimes it's on the capillary endothelium, like it's literally in the blood vessel itself. But and it can be found inside certain cells too, actually, that is true. But it can be found in the blood vessel itself, which I think is pretty interesting. It says not found in the liver over here. That's because the liver has its own enzyme called hepatic lipase, which will break down the triglycerides. So the LPL is found pretty much anywhere else but the liver. It can also be found on the blood vessel walls. And lipoprotein lipase needs ApoC2. That is the cofactor for it. Over here, I told you that lipoprotein lipase, so I told you it breaks down triglycerides specifically. What were the two, I know I've already tested on this, I'm going to test again. What were the two lipoproteins that carry triglycerides? VLDL, Kylomicrons, good. Yeah, so that's why these are the only two that have ApoC2, because they're the only ones that carry triglycerides that need to be broken down. Exactly. Okay, and then you guys already know this. What is the cofactor for lipoprotein lipase? C2, yep, done. And I mentioned this earlier on in the mnemonic, which apolipoprotein inhibits LPL. So I gave you a mnemonic, yeah, that was like ApoC3 lets the triglycerides to be. It prevents the triglycerides breakdown. So ApoC3, LPL is inhibited, which means that the triglycerides cannot be broken down, specifically by ApoC3. Sorry, let me just move the chat box again. Sorry, just give me one moment. I don't know why this is being so glitchy. Okay, there. Okay, yeah, so we've already gone over this, but the function of lipoprotein lipase is that it breaks down specifically triglycerides. I told you that chylomicrons have the highest percentage of triglycerides. Here's something that I want you to know. So depending on what state my body is in, the expression of lipoprotein lipase is going to change, okay? Specifically between adipocytes and muscles. Okay, so adipocytes, when I've just been fed, when I've just had a really high fatty meal, my lipoprotein lipase is going to be activated because all this fat that I just ate, it needs to be stored. And where do I store fat? I store it in adipocytes. So what I need to do is that I need to activate the lipoprotein lipase that's on my adipocytes so that I can break down the lipoprotein and make sure that the lipid is entering into the adipocyte cell. Right? So that's why when I've just eaten a really fatty meal, my adipocytes are going to increase their expression of lipoprotein lipase so that more lipids are going to enter into my adipocytes. On the opposite end of the spectrum, if either I've exercised or I've been fasting, but let's say I've been exercising, my muscles are going to need more lipids. And so they're going to increase their expression of lipoprotein lipase so that they can break down the lipids from the lipoprotein and enter into the muscle cell. Okay, so exercising increases LPL expression in muscles. Eating increases LPL expression in adipocytes. Something else I want you guys to know is that my heart works very hard. Cardiac muscle works very hard. So it has like the highest expression of lipoprotein lipase because it's just constantly working. And so it constantly needs to break down those lipids. But the ones that change by state, by if I've been fed or not, are the adipocytes and my muscles. Okay, so we already talked about this, like the endocytosis and the breakdown from the lysosomes so that I can free the lipids into the cell. And yeah. And I told you that oh my God, this chat box. Yeah, I told you that hepatic lipase is pretty much the lipoprotein lipase version in the liver. Okay, so this is what I explained in the previous slide, and now I want you guys to answer it. Could you explain the relation between apolipoprotein C2 and LPL? Okay, so pretty much on top of imagine a lipoprotein in your head right now, like imagine VLDL or whatever, VLDL or a chylomicron. Attached to my chylomicron or my VLDL, I have the ApoC2. The ApoC2 is going to attach to my lipoprotein lipase. Let's say, for example, this lipoprotein lipase is on my blood vessel, and the ApoC2 is going to activate my lipoprotein lipase. Once my lipoprotein lipase is activated, now the lipids, specifically the triglycerides that were inside of my lipoprotein, can be broken down. So my ApoC2 activates my lipoprotein lipase. Does that make sense? Okay, hopefully that makes sense. Yeah, so it's lipoprotein lipase. Lipoprotein lipase is the enzyme, and enzymes are what breaks things down. So lipoprotein lipase is what breaks down the lipids. Okay, hopefully that makes sense. And then the C2 is just the cofactor. It just activates the enzyme, just activates LPL. Okay, good. So I think I already saw one answer, but could you guys answer this first part? In my fed state, when I've just eaten a really fatty meal, where is the lipoprotein lipase going to be increased? Is it going to be in my, yeah, adipocytes? It's going to be increased in my adipocytes so I can store the fat. When I'm fasting or when I've just done exercise, where is the LPL synthesis going to be increased? Yes, myocytes, muscles. Okay, good job, guys. And I told you that the highest concentration is in cardiac muscle because my heart is working literally all the time. Okay, yes, I know I've already tested you on this. I'm going to test one more time, maybe more, I don't know. But what type of fat is mostly transported inside of my chylomicron? Yeah, one person already answered. Can I get a couple more answers? What type of fat is transported mostly in my triglycerides, okay? And it's from the small intestine onto like my liver. Yeah, so it's really, and obviously it's going to happen after a fatty meal because I told you it's going from my GI tract to my liver. I do want to tell you the small part over here. So I already really nailed it into you guys' head that chylomicrons have ApoB48. They're the only ones that have ApoB48. This is sort of an interesting concept, and I'm going to test you on this again later. But HDL, it sort of like stores these apolipoproteins, apolipoprotein E and the apolipoprotein C, so that it can give them to VLDL or chylomicrons or whatever. So its job is that it carries apolipoproteins on top of it so that it can transfer it to other lipoproteins when they need it. So what they're trying to tell you in this concept is that there are some apolipoproteins that are not transferable. Like they just stick to the lipoprotein all of the time. So for example, ApoB100, it sticks to VLDL, IDL, LDL all of the time. It's never going to be transferred. ApoB48 is stuck to the chylomicron. It's never going to be transferred. But sometimes we can transfer apolipoproteins. Sometimes that little protein that's sitting on top of the lipoprotein, I can transfer it. And specifically, HDL can transfer two classes of apolipoproteins. Either it can transfer ApoE or it can transfer the ApoCs. Okay? I'll say this again later. Okay, so over here, they're just demonstrating it to you. So you guys know that, like, for example, like over here, you can see the ApoC1 and it's just being transferred from the HDL versus over here, the HDL is like giving the ApoC and the ApoE. So the role of HDL is that it just holds them, like it just holds the apolipoproteins. And when my chylomicron needs it, for example, it's going to give the chylomicron the ApoC or the ApoE. So don't worry about like, is the HDL giving it or is the HDL taking away? Because the answer is that it's both. Like when it's needed, it will give it to the chylomicron. And when it's not needed, the HDL will take it away. Okay, so where is the VLDL made from? Like what organ? VLDL was made from an organ. I said it was the mama. Give you a little mama. Yes. Yeah, or hepatocytes. Yeah, like I said the organ, but like the cells are called hepatocytes. And I told you that there's like this little mnemonic, if you want it, that there are 3 lipoproteins that come from hepatocytes: LDL, IDL, and VLDL. LIV, the first 3 letters of liver. They come from the liver. So you can just remember the first 3 letters. Okay, yeah. And then the other thing that I'm going to, okay, I think this got messed up because, yeah, that was supposed to cover HDL, but I'll test you on it again later, so that's okay. But the ApoE and the ApoC, those come from HDL. So you should remember that specifically ApoE and ApoC, they are carried on HDL. What is the function of ApoE? So ApoE, what it's responsible for is that, if you remember, I told you that the liver, it has LDL receptors, right? The problem is that any of the lipoproteins, except for LDL, the liver receptor is not going to recognize it. So ApoE is going to attach to, let's say, VLDL. ApoE is going to attach to VLDL, and then once the VLDL attaches to the LDL receptor on the liver, the ApoE is going to trick the LDL receptor into telling the LDL receptor that, like, hey, I'm also LDL. Make sure that you endocytose me and store me in the hepatocyte. So does that make sense? Like the ApoE is responsible for attaching to the LDL receptor so that lipoproteins can be endocytosed by hepatocytes. They need to be stored or whatever. And ApoC3. So ApoC3 inhibits lipoprotein lipase. So if I'm inhibiting lipoprotein lipase, this means that the triglycerides that are in my, yes, ApoC3 stops the cleavage by lipoprotein lipase. If it's stopping the cleavage, that means the triglycerides are not being broken down. Does that make sense? Okay, awesome. And I told you that there's one protein that I want you to know about called CETP. And essentially CETP is going to transfer the cholesterol that's in HDL, and it's going to transfer it to stuff like VLDL. It can also transfer it to LDL. So CETP is moving the cholesterol from HDL to VLDL. And somebody said before, is it bad or good? It's bad, because we would love for the cholesterol to be in our HDL. We love HDL. HDL is the good guy, it's the hero. Unfortunately, we have CETP, and that's going to move. Yeah, we can take a prayer break right now if you want. Is 10 minutes enough? Okay. It's up to you guys, but I think 2 people said they wanted the prayer break now. We can just stop, like we can come back at 6:20. Is that okay?
Yeah, 6:1 is good, but if we're going to go to the second lecture, we should use the second link.
Oh, we should use the second link if we're going to. We're not going to go to the second lecture though. We're not done with this one. Wait, let me just check how many more slides I have. Oh, actually, JK, I only have like 4 more slides. Okay, I'm so sorry, guys. If you guys are okay with it, I just have 4 more slides. Should I just finish it now and then we join back after the prayer? Okay. Yeah, sorry, that was my fault. I didn't realize I only have 4 more slides. So I hope nobody already went. I'm really sorry. But I just have 4 more slides and then we'll take a break and we'll join back with the second link after this. Okay, so I'll go back to lecture now. Yeah, so I told you the function of CETP. Somebody asked a really good question. It's we don't like CETP because it's moving the cholesterol from sorry, I'm trying to fix my pointer, from the good lipoprotein, which is HDL, and moving it to the bad lipoproteins, such as VLDL and LDL. Okay, so what enzyme did I tell you that breaks down triglycerides is, yeah, what enzyme did I tell you that breaks down triglycerides, essentially? LPL, yes, right? LPL breaks down triglycerides, and specifically triglycerides are found in VLDL and chylomicrons. Good. The liver version of this is hepatic lipase, but it's a little bit different, and I'll go, actually, I'll tell you right now, hepatic lipase is different because it's not going to break down the triglycerides in VLDL and chylomicrons. It's only going to break down the sort of like remnant version of the triglycerides. So remember I told you triglycerides are found in other lipoproteins. So for example, IDL and HDL, it's just not as much. So the smaller amounts of triglycerides that are found in IDL and HDL, they're going to be broken down by hepatic lipase instead of lipoprotein lipase, because lipoprotein lipase acts on VLDL and chylomicrons. So this question, VLDL, I told you is broken down into IDL. What is the last form of that? So VLDL, then IDL, and then what? What is that last lipoprotein, that last breakdown? Wait, somebody said, okay, no, so I'm talking about the breakdown of VLDL. It's broken down to IDL, and then what? IDL, and then what? Yes, LDL, not HDL. HDL doesn't come from VLDL. HDL comes from our peripheral tissues. VLDL is broken down to IDL, and then LDL, yes. And then LDL. And then hepatic lipase, I told you it hydrolyzes triglycerides The remnant triglycerides, small amount of triglycerides that are present in something and HDL. What is the something? What other lipoprotein? Okay, no, it's definitely not chylomicron because chylomicron is not hepatic lipase. It's lipoprotein lipase. Chylomicrons, VLDL, is lipoprotein lipase. Okay, I can't tell if it's an L or an I, but it's actually IDL. And it's because the LDL is so concentrated in cholesterol, it doesn't need, the triglycerides don't even need to be broken down. But the hepatic lipase breaks down the triglycerides in IDL and HDL. So the small amount of triglycerides in IDL and HDL are broken down by hepatic lipase. The large amounts of triglycerides that are found in chylomicrons and VLDL are broken down by lipoprotein lipase. Okay, so hepatic lipase. Could you show the liver mnemonic again, please? Yeah, okay, so liver, so these are the 3 lipoproteins that are made from the liver, okay? And the 3 lipoproteins that are made from the liver, you can remember them from the first 3 letters of the word liver, L-I-V, LDL, IDL, VLDL. So L-I-V should remind you of the 3 lipoproteins that are made from the liver. Does that make sense? Okay, awesome. And then maybe I'll test you guys on this again because I think I didn't explain it too well, but the triglycerides, the remnant triglycerides are broken down by hepatic lipase. The large amount of triglycerides are broken down by LPL. Okay, this is just a diagram, but I think we've really covered it. If somebody asked how C2 and lipoprotein lipase works, over here you can see it maybe a little bit better. Over here, my C2 is attaching to my lipoprotein lipase. My lipoprotein lipase is attached to my endothelial wall. And so the lipoprotein lipase is going to break down the lipids that are already inside of my, I think this is a chylomicron. Oh, no, sorry, it's a VLDL. So C2 activates lipoprotein lipase. My lipoprotein lipase is what breaks down the lipids. Okay, so which lipoprotein did I say was responsible for diet? When you think eating, what lipoprotein are you supposed to think of? Yes, chylomicrons. And what about which lipoprotein was made from the liver? LIV, yes. LIV-VLDL is made from the liver, and it also carries triglycerides. Yes, LDL, VLDL, and IDL all made from the liver. Okay, that's it for the first lecture. Do you guys want to start at 7 for the second lecture? How long of a break do you guys want, like half? Okay, so 7. Okay, we'll start at 7 with the other link. So we should end this meeting, I think, for the second lecture. Okay, hopefully that was helpful. And then you guys can go